Alright, so my phone says it's 12 o'clock, so we can get started. So welcome everyone to the Technology Seminar Series. I think most of you signed in. If you didn't, please do so. It just helps justify the pizza. We just need to count as actual number here. I'll bring the sign-in sheet to the back, so if anyone didn't sign in, you can come over at any point. We do have people watching online generally these days, so if you ask any questions, if you can try to speak loudly, particularly if you're sort of sitting on the periphery. I think the room mics are in the middle and don't always pick up the questions very well, so if you just remember to speak up, that would be. Those are all the announcements I have. So today's speaker is Wei Zheng, and he is a postdoc in DCMB in Yangjiang.
Thank you for the introduction. Today I will introduce our recently developed software called SSER, which try to folding protein by both contact information and threading template information. Here is the outline of my talk. First, I will introduce some background of protein folding problem. Then I will introduce the methodology of the SSER pipeline. Then show some results. Here is the protein structure prediction problem. Protein primer structure is the linear sequence of amino acid, which normally shown by alphabet string, and the local fragment of the primer sequence or the substring of this sequence can form some special pattern. For example, this beta strand and alpha helix in the 3D dimension. This called the secondary structure of the protein. And the entire shape of the protein in 3D dimension space called the tertiary structure of the protein. This fact, the protein structure is a very important key to study the protein function. Actually, it's very time-consuming or expensive to solve the protein structure by experimental tests. So many computer software have been developed to predict the protein 3D structure from the primary sequence. Basing on whether the template used or not, the method can categorize into two groups. One is called template-based method. One is called free modeling method. This slide shows the key idea of threading-based method, or named as template-based method. Given a protein primer sequence, also called query sequence, first the structure features will be predicted by the machine learning method. These structure features include the secondary structure and so on. Then the query sequence, together with these predicted structure features, will be searched against the PDB library, which include all the solved experimental structure. Then it will find the best match, which also called template for this query protein. Then each amino acid in our residue, or called residue in this query sequence, will align to the residue in template structure. Then the coordinate of the residue in template will be copied to the query amino acid as the predict coordinate for this residue. Then you will find in some regions there will some gap aligned between template and query sequence. In template-based method, it will using the a loop fill this gap region. Then in the next refinement step, these two parts will be linked together and remove the clash and so on. Then you will get the final model like this. There are some several very popular used template-based methods. For example, SSER, Lomas, and HHP. From this pipeline, we can see the template quality can decide the template-based modeling model quality. So if you can't pick up the correct template for your query sequence, or the template, good template, not exist in the PDB library, generally you will the template-based method will fail for structural modeling. To address this problem, people develop the free modeling method, also called AB initial method. In AB initial method, they want to code the protein from scratch without any information from the template. So they will build the initial confirmation with some random loop, then folding this protein along with this free energy surface to get the minimal value. Then you will get a predict model. Here the free energy can be either knowledge-based potential or physical-based potential. Although the so there are several popular used AB initial methods. For example, Quark and Rosetta developed by David Baker. Although the motivation we developed AB initial method is very good. We want fold without any information from template. But due to the free energy is not so accurate, actually the free modeling method or AB initial method really not good as with the template-based method due to these two problems. But things has been changed after a concept that called residue contact has been introduced to the AB initial modeling method. This figure shows the definition of the concept of the residue contact. Given a protein's 3D structure, if the C beta atom distance for this residue I and the residue J is less than 8 angstrom, then these two residues form a residue contact. And actually, if we label this protein residue from 1 to L, from the protein N terminal to C terminal, then we can using a L square map or matrix to describe the protein residue contact information. Here X and Y both are residue index, and the black point means the residue contact, and white point means non-residue contact. For example, this point means residue 55 and residue 105 form a contact. Then from this definition, we can see actually the contact information is not useful for the protein folding problem, because you must know the 3D dimension structure, then know the contact map. So unless you can get this contact map from protein sequence, this feature is not useful for the structure prediction. Recently, research, people find the residue which forms contact have some special relationship in the protein evolution. This special evolution called coevolution, which means they found the residue which forms contact. They usually mutate together, and this coevolution can be observed in the multiple sequence alignment for this query protein. So after people notice this feature can use for predict the contact, residue contact, several machine learning methods have been developed to predict the contact information from the protein primary sequence. After this residue contact become predictable, this information has been shown very useful for protein structure prediction. For example, if we can predict these two residue formed contact from this protein sequence, then during the protein simulation, we can pull these two residue close together to form a contact. Then this contact restraint is very directly information to help you fold the protein structure. And this figure shows the comparison with the AB initial method without contact information. We show the quark result and the template-based modeling method at SSER, and the AB initial method with the contact as additional restraint. Here, Y is the TM score, which is a measure for quality your model qualify your model quality, and the higher TM score means you have the better model. So from this figure, we can see without the additional contact information, AB initial method quark actually not good as with the template-based method. But after we introduce the contact information to quark, then the C quark is much better than the template-based method at SSER. So this proves the contact information is actually very useful for the protein folding problem. Then we think whether we can introduce this contact restraint to the template-based method to improve the performance of SSER. So this figure shows the key idea of why we want to introduce the contact information to the folding problem. Sorry. Given a protein sequence, so we can first using threading program to pick up the template for the query protein, which show in this figure. The template shown by the blue color, and the experimental structure shown in black color. We can see the local fragments or the local structure that the template can fit well with the experimental structure. But we can see the orientation of this helix is much different from this experimental structure. This means your template is not good enough. But if we can predict the contact information between this helix and this base strand from the sequence, then during the folding simulation, we can pull this helix to form the contact, to close to this base strand, to form this predict contact. Then you will get the final model, which showing the red and yellow color. So this is the key idea that why we want to introduce the contact to the template-based method. To implement this idea, we build a pipeline of CI tester, which showing in this figure. Starting from the query sequence, we can first using the meta-threading server called Lomis to pick up templates for next step. In the same time, we can using MSA multiple sequence alignment generation software called DCMB to build a multiple sequence alignment for this query protein. Then this multiple sequence alignment will feed to a set of contact predictors to predict the contact maps. Then the information from this template and contact maps can be used to get the folding simulation to get the to sampling the structure confirmation or decoys from this simulation. Then the decoys or the confirmation from this simulation step can be used for spectral cluster based on the structural similarity. Then the cluster center of this largest cluster will be selected as the initial decoy, initial model for the refinement step. In this step, a software called FGMD will implement to the initial model to remove the clash and add the side chain. Then you will get the final model. In the next slide, I will show some details of the CI tester pipeline in each component of the CI tester.
So, quick context question. So, DeepMSA is the Google DeepMind entry, or is it something else?
No.
It's something our lab developed.
It's all, there's no Google. This is like Google grad versus this. This is the competition to Google. This
All right.
is Google.
So there's no Google chunks in here.
No.
Like, so just for, so is this the same CASP competition that Google won? Or
It is,
the
but
upcoming CASP.
But it's Google won one entry for the machine learning last year. These guys won it for
I know
7 years.
So yes, so this,
and they
this
won the other competition with iTaser and then Quark last year. Machine learning version, they were second. Okay, you got to get it out there.
Yeah, no, I know.
I'll tell you why we got to get it out there, because the press picks up on Google and they don't pick up on these guys. Okay, and you know, if we had 15,000 processors, you know, it might be a different story with Google. That's part of the problem. It's asymmetric warfare.
So,
say in the Defense Department, right? With me?
I will introduce some detail of the DeepMSA pipeline in the next slide. This slide shows the threading method implemented in CI tester pipeline. So, since we noticed the single threading server or single threading method will have some advantage and disadvantage. So we implement a meta-threading method, which include 11 single threading methods to send the templates for CI tester. Starting from the query sequence, this query sequence will pass to 11 individual threading programs to send the template from the library. Then you will get a set of templates from each individual threading. Then we will using Lomis to re-rank this template, and this re-ranked template will be as the initial confirmation for the next step, simulation step.
I have a couple of questions here. Have you been able to kind of figure, there's several, this is fascinating. Have you been able to figure out, like, when you run one protein versus another, do the success of the different programs change? I mean, is there one program of these 11 that's better than another all the time? I mean, how does that work?
Actually, when we benchmark the results, in some protein, this program may be better, but in some other targets, this program may be better. So that's why we using the meta method to combine the advantage of different single threading methods.
So the question is, I mean, it's like the one that CNF search. Is there a certain kind of protein where that works better? Like say a alpha barrel or something. Some template where that works better than then you have a beta sheet versus something where you have some flexible. I mean, have you been able to figure out which program works with what kind of structure better? It'd be interesting to see that. That was one kind of question. Another one is, how do you manage that computationally? So are there different processes that are going? I mean, how does the workflow computationally? I mean, does it take different amounts of computational power to run these different algorithms, and then you collect the results, you run them independently, and then you collect the results and then pool them? I mean, how does that work? Eric, you might know the answer.
Yeah, so each one of these threading programs is its own job on the supercomputer. Obviously, they're going to be very different in terms of how much time they take.
Yeah, that's what I was wondering.
But all of them are going to take maybe a couple hours at the most. Threading is pretty quick.
That's the time scale? A couple hours.
Yeah, for template identification, that's about what you're looking at.
And how many processors and how much memory is required for each one of them?
Memory is dependent on how long your sequence is. Processors you only accept single-threaded jobs, so these are all single-threaded. Other independent jobs, which is where the parallelization comes in.
Yeah, so it's embarrassingly parallel, and you got 11.
Yeah.
So, well, is it more parallel than that? So, do these take the entire sequence as an input, or do you chop it up on fragments and then feed the fragments to each?
I don't know. Are you going to talk about domain division, or is that something?
Yeah, that would make sense.
Actually, for the single domain protein, this is a good problem. So, for the single domain protein, we didn't cut the protein to the segment. We just using the entire sequence as the input. But for the multiple domain proteins, actually we using some software to cut this protein to different parts or different domain. Then the domain level sequence will be put to this long threading method to pick out the template. And further to the modeling step. And after we get the individual modeling of each domain, we get the model of each domain. We using another software can merge these two domains together. And this have been proved the accuracy of the final modeling is better than if we put the entire sequence without domain split to this entire pipeline. Mm-hmm.
That makes sense, because most of these programs would then try to make it into a single.
Yeah.
So I know at some point, I apologize if I'm skipping ahead, that you chop these different templates up into pieces, and you can use some fragments from one template and some fragments from another. Is that what these lower steps here are on this slide, or is that going to be downstream later? So you might, as I understand how this works, you could use the first 80 residues from F search and the next 80 residues from SP3 as your template going forward. You don't have to take the entire template as it. Is that not true?
That's actually not true. So the entire sequence will put to each individual threading, and which different threading may have different algorithm. Some may pick up a very long coverage template for this query sequence, and some program maybe pick up some short coverage template for this query sequence, and all this will be merged together. So there is this doesn't mean the first fragment will input in this threading, and the second fragment will input to this threading. So this we actually put the entire sequence to each threading program and get the different template.
Although I think the case that he's talking about is the case in which one program identifies the first 80 residues are well aligned to one template, and then another set of the residues are well aligned to another template.
Right.
Mm-hmm.
Rather than, so in that case, yes, you will then combine those two separate pieces of information into the folding system.
That makes
But
sense.
it's not as if you split apart the fragments and then put one fragment into every threading program. All of them are threading the entire sequence.
This is, correct me if I'm wrong, this is just essentially giant ensemble.
Yeah.
Prediction.
Yeah.
Yeah. That's all we're doing. It's just ensemble.
It's just consensus prediction.
Seeing what the most likely domain is and selecting it based off of those specific, all the output of all those different algorithms. Saying, based off this section, the most likely prediction for this domain is this. Based on this section, the most likely domain is this.
Yeah.
Is that correct?
Yeah. Any question?
So I guess the question, based on what Marcus was saying, is how much different are the outputs from these different programs? How much different are they?
Not for it to matter.
Yeah, but that's the qualitative answer, and it's somewhat acceptable. But what you'd like to know is some scoring system.
And it would be nice to know if there were meta information about the target that could tell you which of these. I mean, I assume that it's more likely to make a good guess at it for whatever reason it thinks. But, beyond that, the high entropy structure that you're trying to predict, is it eukaryotic versus bacterial? Make a difference? Probably does.
Trying to check the function of the templates, and we rank the template based on the function values.
Yeah.
It ends pretty badly. Function is much less conservative structure. So
Yeah.
trying to re-rank the template by functions varieties usually not.
It's pretty interesting. Something deep in that. Yeah, that's interesting.
Okay, then we will continue. Here,
Okay, good.
we show the benchmark result of the Lomis result, and its competent 11 single threading server. So Y is the TM score of the first template. So we can see for meta-threading method, Lomis is at least 10% better than the second best method, HHPRED. So this means actually, if you're using meta-threading method, it's actually improve the template identification ability than single threading server. Another very important component of the SSER is the contact prediction programs. Here we show a pipeline of the contact predictor, which named as triplet RES, which developed by our lab member Yang Li. Still starting from the query sequence, we will first generate the MSA multiple sequence element, and the three kind of co-evolution features which will be calculated directly from this multiple sequence element, then pass to the deep residue networks blocks, then generate three feature maps. Then in next step, we will merge this feature map in one feature map, then pass to this deep residue network blocks again, then get the final predict content map. Similar with this idea of using meta-threading server for identify templates, we're also using 13 individual contact predictors in CI tester pipeline, and this figure show the benchmark result of these contact predictors in CI tester. Here, red bar means the contact predictor developed by our lab, and the star mark means those predictors are using deep learning technology. Otherwise, it's not used the deep learning. So from this benchmark result, we can see the method using deep learning training or deep learning technology is much better than those methods not using deep learning method.
So it's about twice as good as Gremlin. Is that an area under the curve for successfully predicting a true contact? What is that number?
Probably precision, right?
Yeah. So this is the y-axis is precision, and why we introduce some lower accuracy or precision method to CI test pipeline. We found for some special targets that Gremlin or CCM still can generate some correct content map for this special target. So that's why we try to using the meta method similar with the threading pipeline.
Are all of these methods running on the same multiple sequence database backend?
Yes.
So they were all trained, and when you trained all of these, you were training them on the same PDB structures. X. For the PDB?
Actually, the training pipeline, actually they made training by different experimental structure and different MSA, because some methods are developed by other groups. But in our using, we actually using the same MSA input this all individual contact predictors.
Okay, so they're all getting the same input, but they were trained
with
not
the
necessarily
difference.
under your control.
Yes. Since we may notice this MSA is very important for the context predictors, so we build a new multiple sequence element generation software by our lab called the DCMB, which still combine a set of single homology sequence detecting tools, include HHBLIS or JCamera. They are single homology sequence detecting tools. We're using this combination of the tools, searching with the regular genome sequence database and meta-genome sequence database. Here, compared with the regular genome sequence database, meta-genome sequence database may contain more sequence, which provide more chance for your query sequence and detect more homology sequence for your final multiple sequence element. And we using the contact predictor to benchmark different the quality of MSA generated from different pipelines. Here, the red bar means MSA generate deep MSA, and we show some control method that MSA generates from the single homology sequence detecting tools. We can see for these 5 contact predictors, if we using MSA generate from this deep MSA pipeline, the performance is much better than other MSA generating software. This means our pipeline works better than other software for those contact predictors. After we getting the template information and predict contact map, we using Monte Carlo simulation to sampling the structure confirmation or decoys for this query protein. So this pipeline is a very typical Monte Carlo simulation pipeline. Here we're using the initial confirmation from the Lomis threading. So we'll copy this aligned template as the initial confirmation of this pipeline, and using the movement implied in the CITES pipeline, which show here, to change this confirmation to a new confirmation. Then we will basing the metric policy rules to decide whether we accept this new confirmation or reject this new confirmation. The metric policy rule show here, which you need to calculate the energy for in CITES, which I will show the energy in next slide. After the m times circle in this simulation, we collect the accept confirmation or decoys to this confirmation pool. Then all this confirmation will be used for generating the final model. As we mentioned in the last slide, we need to calculate the energy to get your Monte Carlo simulation. Here in CITES, we have 4 kinds of energy potential, which the highlighted by red is the contact potential, which we introduced newly to the CITES pipeline. This is the equation of this contact-based contact potential, and this curve shows the function of this figure shows the curve of this this equation. We can check this curve. The x is the distance between residue I and j in your confirmation or in your decoy during the simulation, and y is the energy values. For example, if you predict residue I and j should be forms contact, but during your simulation, your decoy, residue I and j are not formed contact, so your energy will be the energy of this decoy will be very high. Then pushed by this energy function, these 2 residues will be pushed together to form a new decoy or new confirmation, which these 2 residues can form the contact. Then the energy of this decoy should be get lower in this place. That's why we design energy potential like this to form to get our Monte Carlo simulation. Actually, when we're using single Monte Carlo simulation, there is very easily to get a local minima, because if you set the temperature in the Monte Carlo simulation too low, then this will be very easily to track to some local minima, can't get the global minima. So actually, we using the replica exchange Monte Carlo simulation to solve this problem. So we taken n different replica of the Monte Carlo simulation with different initial templates, which coming from low-mid threading and with different temperature setting. After m times local movement by SSER, then we will swap the confirmation in the neighbor two replicas. Then starting from start to next circle of the Monte Carlo simulation. At final, we will collect all confirmation or decrease generating from the lowest 5 temperature replicas. Then those confirmation will be passed to the SPECR program. SPECR will try to cluster this confirmation basing on the structure similarity between each device. And the cluster center of the largest cluster will be selected as the initial final model. Then, since this model will only contain the backbone, so we will be using the DCMB program to add the side chain of this protein and remove some clash and so on. So then we will get the final model. So this is the entire pipeline of the SSER. Includes threading, contact predictor, Monte Carlo simulation for sampling device, and SPECR for cluster the device, and FGMD for refinement the final structure. And here is the benchmark results of the SSER versus SSER. SSER is a pure template-based method, and the SSER interplays both contact information and template information. Here, each point means one protein in our benchmark data set, and blue point means the target protein that you can easily find some good template. And the red point means those proteins you can't easily find or you can't find the good template. So from this benchmark result, we can see our new program SSER is much better than SSER, especially for those targets that you can't find good template. So this emphasis indicates the contact information actually works very well in the SSER pipeline.
So are these targets that you're counting more or less non-redundant?
Yes.
So there are no two targets are high.
Yeah.
So this is a big enough data set that you could answer some questions. You know, is it working better for bacterial proteins or eukaryotic proteins? Working better if they have a larger hydrophobic amino acid content? You know, I mean, 300 dots is a fair number of dots, even in that hard category where you really care. But one very basic, what I think of as a very basic question, is hard targets where you're getting a lot of improvement when you incorporate these contact map predictions. Do they have a deeper MSA all the way back when you tried to predict those contacts? Not true.
I mean, you could probably answer it better than I could.
So your question is those hard targets.
Hard targets that improve a lot.
Yeah.
And so there are hard targets that improve a lot, and there are hard targets where your prediction is hard targets where your prediction is still bad. Is it just that you don't have enough homology model? You don't have enough homology sequences in your MSA to predict the contact, or is it some other problem?
So actually, for those hard targets, they still worse than the easy target. There are some key problems in this. So one is you can't find this good template. Still, this template information is not good as this easy target. Another is actually this contact accuracy is not good as with those easy targets. That's what we observe in the benchmark data set.
That's not a surprise because the more prevalent something is in sequence space, the more likely it is for something to crystallize it and have a good template. So in fact, Protein Structure Initiative, when it was running, was systematically going after targets that had a lot of homologs, crystallizing. So anything that's an easy target because there's a PSI homolog feature solved is almost certainly also going to be something that's going to have a deeper multiple sequence and therefore better contact.
Actually, the contact performance and the template performance are very related, since the performance of contact predictor depend on the MSA generated from the homology sequence, and actually threading program which also using this MSA to generate some profile. Secondary structure prediction or profile is a concept also generated from the sequence element, multiple sequence element. Nearly almost threading using this profile concept to align or pick up the template from the library. So the multiple sequence element is a very important things in both threading and contact. That's why for the hard target, if you can't find good template, actually you can't generate good contact either. That's why.
And also, it should be stated that the correlation between contact prediction performance and number of homologs is in fact there, but A, it's not as tight as you would think, and B, the correlation stops after a certain point, meaning you get diminishing returns if you have enough homologs. That's why in our deep MSA pipeline, it's possible to stop at either column 1 or column 2 if you have enough homologs. That going deeper into the multiple sequence alignment is not necessarily going to give you better performance. And in fact, in some cases, will give you worse performance because it'll introduce much more noise.
Yeah. That's good.
Okay. So this is our server performance in Casper competition. So critical assessment of protein structure prediction is a worldwide experimental experiment for protein structure prediction. So the organizer will release some sequence to prediction groups and experimental groups. Then prediction groups will solve the predict the structure model, and experimental groups will solve this protein structure by experimental tests. They work independently. After several months later, the organizer will collect the model from prediction groups and the experimental structure from the experimental test groups. Then they will compare the model and the experimental structure and the ranking the partition groups, see which groups performance well in the prediction problem. So in the Casper 13, our CI-tester server ranked as the first top one ultimate protein folding server, following by the CQuark, which is another contact get protein folding server developed by our lab. So this is the performance of our folding server in Casper experiment. And it's very meaningful problem that what kind of protein can foldable are foldable by CI tester. Here, foldable means the foldable that if the CI tester model have 10 square greater than 0.5, then that means this target foldable. Here, we using this figure to answer this question. Here, X is the performance of your contact predictor. Y is the performance of your threading template. We can see in these two subregions, that means you have good template. And in these two subregions, you have good contact. That means either you have good template or you have good contact. The protein is foldable. But very interesting is in this subregion, neither you have good contact nor you have good template, but there's still like 30% protein still can foldable in this subregion. So we are using this case to highlight why there are some proteins still foldable in this subregion. So when we check the contact map, the green point is the contact from experimental structure, and the red point means predict contact. So if these 2 points overlap, that means correct predict. Otherwise, it is wrong contact. So when we check the contact in this region, we can see almost these contacts are wrong contact, which means this contact map is not perfect. When we check the template from the threading, which show in this figure, the template highlighted by the yellow color and the gray color is experimental structure. We can see, although this base spread align very well to your experimental structure, the orientation of these two highlights in your template is still wrong. But when we check the contact map, we find the contact between this helix and this base spread is correct predict from the contact map. So during the folding simulation, we can pull this helix close to this base spread to form the contact. Then you will get the blue final model, which is much similar with your experimental structure than your initial template. So this case shows the power that using the both template and contact information to get your protein structure prediction. Since time seems okay, we can skip this case to conclusion. So we have developed a new protein folding program called SSER, which interplays both information from threading template and predict contact by deep learning technology to get the Monte Carlo simulation to fold the protein. Based on our benchmark, SSER is at least 46% better than our former folding program at SSER. That's all. Thank you.
Since I have a lot of time for questions.
Okay, so
if there are no more questions.
Good idea.
Yeah, so
he wants to go
cover
to
the biology stuff.
this one.
There you go.
Here we show one case to indicate that CI tester can help biologists to solve the biology problem. So we know one protein complex called MLL complex, which is an enzyme that can regulate the gene expression. So that means we have a protein complex that can bind to the DNA molecular. But although each component of this protein complex have already solved, but the entire complex, that means how each individual monomer of how this monomer forms the complex to bend this DNA molecular. This is still unclear.
Right, so that's the nucleosome 4 particle.
Yeah.
Just an H3H4 tetramer in there and some DNA wrapped around. That's fascinating. So what have you been able to figure out?
So one of our collaborators, they want to know how this protein forms the complex. So they solve the because there is currently no software can solving this huge complex. So
Right.
they solve the core-EM density map for this entire complex. So you can imagine this core-EM data like some shape of the entire complex. Then the idea of they solve this crystal structure, they want fitting each individual monomer structure to the density map to this shape. Then you can get guiding by this density map, you can get the correct complex and find the interface between protein and the DNA molecule. But one problem is one component of this complex, ASH2L, they don't have the crystal structure. So we're using our SSER build the model for this protein. And they using the solved structure and our model fitting each component to this core-EM density map, or you can imagine the body or shape of this complex. Then they get this protein complex like this, and they find two binding reasons between this protein complex with this DNA molecular. Especially one binding reason is just located in the ASH2L model with this DNA, which show in this figure. And we calculate, basing on our calculate, we find 6 residues are close to this DNA molecular. So our collaborator, they do some experimental tests. They try to mutate this those 6 residues to other kind of amino acid. Then they found after mutation, this complex will lose the binding ability to this DNA molecular, which shows the 6 residue we found in this in our predict model actually is the potential binding site to this DNA molecular. So this work is published in this journal last year. So this case just indicates our SSER model can help biologists to solve some biology problem. Any question with this?
That's very interesting. It shows what it does is it would disrupt the nucleosome. It actually would disrupt the whole, 4 nucleosomes. Actually, it sits between the nucleosomes, so it's gonna really open up the chromatin structure in a way we hadn't thought of before. It's very interesting. You have a very good example. I'm glad you had a chance to show.
So was there any energy minimization done after the superposition step, or is this just straight up a monomeric model superposed into the EM density?
So you mean in the fitting step? Or?
So was there any minimization to optimize how the interface was contacting DNA? Or was it just that we modeled it monomerically and plugged it into the cryo-EM density and just used the interface that arose as a result?
So actually, we just using the raw model fitting. In the fitting step, the experimental structure will do raised-body fitting. They don't change the conformation. For the model, we using the flexible fitting, which means the interface may slightly change compared with the initial raw model. And this work is done by Bill, and For the interface residue, we just calculate the distance between the side chain residue with the atomy in this DNA molecule. So we didn't do other things.
Although if I recall, Yao's program is just meant to fit the protein density into cryo-EM density. Fit the protein structure into the cryo-EM density. It doesn't actually take into account energy of any sort, right?
Mm-hmm.
So I wonder how this would change if you tried to use like a more physical force field.
Well, we know its resolution cutoff area is about 8.
Right.
I mean, it's pretty low res.
And I mean,
But those contacts are interesting and could make a difference while you're talking. My guess is, just as they did with the ribosomes, that this is the beginning of a process similar to what you're suggesting, and would be a good refinement. Yeah, very good. My guess is that it's just fitting the density right now, but it's pretty cool. You can see where, there is some room for improvement up at the top near the ASH2L that it's set. But it's really pretty impressive. It's only going to improve. And it's already telling us things. I mean, and it's really certainly more informative than just like blobs from cryo-EM. Really good.
Any last questions?
Thanks, Sam, for having me.
Thank you.
Very impressive.
I was the only one who would